Alright, so I apologize about the delay in this video. Just been a little busy at home, lots of things going on, and an extra long commute home yesterday. So I didn't get to make it on time or even by my delayed deadline. But we still have enough time to get this done by Friday's class, so we'll still make it do. Alright, so I'm going to talk pretty quick because there's a lot to go over, but a lot of it's review. So what I'm going to review is the enzymes of the bacteria and the activated sludge. So I want you to flip back through your notes and look at the wastewater treatment notes and the sewer science information to review what part and what stage of the filtration system activated sludge fits into. I want you to recognize that there's bacteria in that activated sludge, despite the fact that you were not looking for bacteria in your microscopes when Miss Melissa came to our school and you were looking at the activated sludge with a microscope. You were, instead of the bacteria, you were looking for protists and animals, not bacteria. You wouldn't have been able to identify the bacteria. They're way too small. If you remember, like compound microscopes don't give us a lot of clarity on the bacterial cells. And so what you should know is that this activated sludge is a mixture of microorganisms which contact and digest biodegradable materials from wastewater. So there's a lot of loaded information in that definition that we need to make sure we understand. It's a mixture of microorganisms. I hope you can identify which type of mixture. I'm not going to tell you, but I'm telling you that you need to know that it determine whether it's a solution, suspension, or a colloid. We're going to review what microorganisms are and what these biodegradable materials are and how these microorganisms break those things down, how they go through catabolic reactions. If you don't remember what that is, review your notes from an earlier video or from last year. This is a biological process, which I was saying in class, which I think is really cool, that we're able to take something from nature that has evolved, that is the product of this long process of natural selection, to give us microorganisms that are functional in this environment. But we're able to optimize it, which is a really kind of interesting thing that humans can do, is manipulate the environment, find the optimal variables, environmental variables for these bacteria so that they do the job we want them to do really, really well. So if it's a biological process, we must therefore properly control the activated sludge process. And in order to do that, we must control the growth of the microorganisms. So we have to control how quickly they grow, therefore how efficiently they can be doing their job of breaking down organic compounds. So to control the growth, what we need to be doing is controlling different factors and variables that affect those microorganisms. So you know that aerobic microorganisms or aerobic bacteria need oxygen, and so you saw them bubbling. The bubblers also, in addition to adding oxygen to the environment and bringing those microorganisms in contact with the oxygen so that they could do aerobic cell respiration, make lots of ATP, grow, process well, break down food, get larger, divide, make more organisms. It also mixes up all of the nutrients. But if you bubble it too much, then some of the flock or the clumps of material actually break apart again, and then it actually reverses what you're trying to do. So there's an optimal amount of bubbling as well. But some organisms, like the anaerobic bacteria, don't want that bubble. When we're talking about these microorganisms, what we really need to talk about is that 95% of those microorganisms, or about 95%, are made up of bacteria. You weren't looking for bacteria again through the microscopes, you were looking for larger forms of life. But bacteria make up a vast majority of that activated sludge. And because those bacteria are microorganisms and living, what they're doing is consuming biodegradable materials such as carbohydrates, lipids, proteins, and other compounds. So organic and inorganic compounds. So it's important to understand that, review carbs, lipids, and proteins, how they affect, how we can break them down and build them up. Remember your hydrolysis and dehydration reactions. Relate that anabolism and catabolism, and know your examples of carbs, lipids, and proteins. That's all wonderful stuff to review, and I'm asking you to do that to prepare for an upcoming assessment. Okay, so how do these microorganisms do that? Just like anything else, enzymatically, right? So biological processes are often catalyzed, right here. So enzymes are proteins that catalyze reactions. They speed up the rate of the reaction by increasing the affinity. I'm going to add a little note here. That just means the attraction. Affinity is kind of a way to think of attraction of the substrate and the enzyme. So review your notes on enzymes. Remember what an active site is and what the substrate does at the active site of the enzyme. But it catalyzes the chemical reaction by attracting the substrate and the enzyme together. That's a critical piece of information to know. And so remember that if enzymes are proteins, right? Enzymes are proteins. These proteins are sequences of amino acids. Proteins are the macromolecules, and amino acids are the building blocks or the monomers that make those proteins. But the order or the sequence of them are critically important to make a protein function correctly. So the sequence of those amino acids, which are assembled by ribosomes, remember they're either free-floating in the cytoplasm or in the rough endoplasmic reticulum, are determined by the sequence of mRNA, messenger RNA. So mRNA is coming to the ribosome assembles the amino acids in the correct order, it utilizes another RNA, I hope you remember transfer RNA, look that up. There are codons and anticodons involved in that process, and that the mRNA is determined by the sequence of DNA. So DNA leads to RNA, which leads to the protein sequence or the amino acid sequence. That's going to be a critical piece of understanding when we get to some data to look at for support of the theory of natural selection. The sequence of DNA, if you remember, you get that, or these bacteria get it from the previous generation. So bacteria don't usually go through what we would call sexual reproduction. They just divide and they make clones, but they do that so quickly and so many times that their DNA or their genetic code is vulnerable to mutations, and that's how they get variation in their population. So that's a quick overview of enzymes, proteins, and where this all fits into the whole process. So what I'm going to do is add one more thing here. Oh, sorry. For these enzymes, I forgot about this piece right here. When we think about enzymes, we need to think about those optimal environmental conditions. So think about what kinds of things affect the rates of enzymes and what causes them to denature. You should review your graphs on pH, temperature, and substrate concentration. And know what's happening to enzymatic rates of reaction under those different environmental conditions. Okay, so the next thing that I have here is this graph that I sketched out. So we're going to talk about the role of enzymes. You did not see this graph last year when we talked about enzymes, so I want to make sure I take a minute to explain this. On the x-axis, I've got the extent of the reaction. So these are just lined up. It has nothing to do with the overall speed of the reaction. It's just saying from start to finish, and that's scale to start to finish. It does not indicate that one is faster than the other. What this graph does instead is tells you the amount of free energy or the energy that's required to drive a reaction forward. Here are the reactants of a reaction. Here are the products, right? We start with reactants. It goes through some kind of chemical reaction, and then we end up with products. What I've added here is this horizontal line that's going to indicate our initial amount of energy. And in order to drive the reactants to the products, we need to add energy. So the amount of energy that's required increases. So this is energy added above this dotted horizontal line. So there are two graphs here. So I'm going to tell you that, let's start with the orange one. Maybe I should do that in the color orange. Orange is going to be the uncatalyzed. It's going to represent the uncatalyzed reaction. So it starts here, they both start at the same amount of energy, but overall more energy is added to the reaction in order to drive the reaction to the same products. If you notice that the distance between the original amount of energy and the peak of this orange graph is far greater. That's the amount of activated energy or activation energy required. So that is activation energy. So the amount of energy that's required to activate a chemical reaction. But with an enzyme, because of its chemical properties and its ability to bring a substrate and enzyme together, requires far less activation energy. So what we have here in blue is the catalyzed reaction. Okay? So catalyzed reactions with an enzyme will reduce the amount of energy required for the reaction. When there's less energy required, there are a couple of benefits. So it increases the speed of the reaction, and it reduces any heat that comes off the reaction. So your body doesn't overheat. Your body could still process carbon compounds and anything else without enzymes. But the amount of energy that's required is far more than you have, and the amount of energy that comes out of the reactions is far greater than your body would be able to process. In fact, it would denature other proteins that are not enzymes in your body. And so this difference where the products are from the original amount of energy is the change in energy. And we have a negative change in energy. There's a loss of energy, and that loss comes out as heat. Okay? So it's an exothermic reaction when that energy is lost. All right, so let's talk about this very quickly, the enzyme reaction model. This is just a word diagram that we talked about last year, and this is really kind of just describing the idea that enzymes are recycled, right? So they're reused over and over and over again unless they're denatured. And in which case they might get recycled as well because they get chopped up for pieces, like in a chop shop, like a car in a chop shop, and it gets reassembled. So we've got an enzyme, which is a protein, and some kind of substrate that it's going to work with. They will combine in an enzyme substrate complex, which is when the substrate fits into the active site of the enzyme, and then after the reaction, we end up with an enzyme and a product. And then that enzyme can be recycled over and over and over again. So as long as you have sufficient amounts of substrate, this reaction can continue at increasing rates. But if you have too much substrate, the substrate's just going to be waiting around until the enzyme is available again to take the next molecule. It's like you're waiting for someone to walk with you to class, but that person has to walk somebody else to class first, and then they come back and get you. If you have too many people waiting, the reaction doesn't go fast enough. Your body could make more enzyme if it gets the chemical signals to do so, which would speed up the rate of the reaction, which sometimes happens. All right. So the last thing that I want to talk about is the growth curve of bacteria, and this connects to what you've been doing with Mr. Canales in math. So what I've done here is drawn a graph that shows the growth curve of bacteria. On my x-axis I've got time, and then on my y-axis I've got biomass concentration. And what I've done is actually put these green horizontal lines. These are not asymptotes. These are just markers to break up this graph into something we call piecewise functions, right? So we can talk about each one of these phases separately. So what I'm going to do is put in my terms here and explain to you what's going on. And this is not drawn perfectly well. It's not a perfectly well done graph, but it's a sketch of what that graph would look like. And I think that you'd be able to do some calculations on this and some math, which I would hope you are able to do on a science assessment. You can pull in that those growth factors that you have to kind of analyze a graph a little bit. That's what I would expect you to be able to do on an assessment, on a benchmark. So what we have here is an initial amount of bacteria. In the exponential growth phase, the rate of growth is increasing. The rate increases, so it gets faster and faster and faster exponentially with growth factors. And so what's going on here biologically is that these bacteria have lots and lots of resources available to them, and they are able to divide pretty quickly and increase the population. Now, I don't have number of individuals here on my y-axis. I have biomass concentration, because it would actually in reality be very difficult to count the number of bacteria, but you could take the collective mass of the bacteria. And that collective mass is increasing very, very quickly. And then at some point there's something called an inflection point. There's an inflection point right about here where the rate changes. It actually moves to declining. The rate starts to change and lower. It's still increasing, right? We're still growing, but at a slower rate. And then we end up at a stationary phase. But the declining growth phase is because there's less and less resources available. And at the stationary phase, all the resources are being maximized out. In the endogenous phase, the death rate is increasing. And this is not what you would want in a wastewater plant. This is not what you would want when you're trying to manage the bacteria. But when the resources have run out, the bacteria are simply trying to survive, but there aren't enough resources, so there's a death rate that would continue to extend downward. And if there are no more resources, or if they're unable to access the resources, either because of environmental conditions have changed, the enzymes that do all the work for them are no longer functioning, or there are molecules that they can't break down because their enzymes aren't structured, the active sites aren't structured to actually interact with those molecules, the rate would come crashing down. Okay, I see that I'm at 16.5 minutes. I'm going to stop right there.